So we stay with fMRI, and our next speaker is Grandjean. He's doing mouse functional connectivity studies at the Donders Institute at the Radboud University in Nijmegen in the Netherlands. And he's working hard to make rodent fMRI work in multicenter studies. Today, he will tell us about the lessons learned from working together as a community in these studies. So please, Joe, go ahead.
Thank you, Claudia. It's my pleasure to be here. As it's been pointed before, I think it's my third year in a row that I'm presenting, and it's always the same project, but it's evolved over time. And today, the way I want to present it is not as a scientific talk, but properly as propaganda. It's also a love letter to the neuroimaging community that I'm part of. Everyone who grows up in science have these kind of papers that, you know, when they do their PhD, it blows their mind and it motivates them to carry on their research. And for the purpose of that talk, I love to share to you two of my favorite papers of all time. The first one is from Drevets, 1997. It's actually before functional MRI, and they used nuclear imaging to look at metabolism in the brain of participants with depression and without. And they found that in the brain, there was a hot spot where there was hypometabolism in participants with depression. And that's fantastic because it was the very first study that could show that in psychiatric disorders, you have something biologically happening in the brain, a biological manifestation. And I find that fantastic. The next study I really love is one by Filippini. And there what they did is they looked at carriers for a risk allele to develop Alzheimer's disease. But they look at participants when they are in their 30s. So cognitively healthy, no problems at all. And then they run task-free fMRI on them compared to participants who don't carry that risk allele. And they found that already at 30 years of age, 30 years before developing Alzheimer's, potentially, there was already differences in the metabolism and the neuronal activity of the hippocampus, which is the region that encodes memories. And that's the first to be hit with Alzheimer's. And that's fantastic because it really shows the potential for functional imaging to really understand brain disorders and to try to capture them as early as possible. I'm not the only one who's been excited by it. It led to an entire community branding together and putting all the resources to get these fantastic data sets that are now sort of the bread and butter of human neuroimaging studies, starting with the Human Connectome project that has about 1,000 MRI scans, UK Biobank being the largest with 50,000 MRI scans plus genetics. But in contrast, if we look at the animal imaging community, it's like we're a small fraction of that. In a previous study, we counted the number of fMRI studies in animals. And here, this is a representation. So we have a lot of rat fMRI studies, and mice is actually growing up. And then we counted the number of papers per year. And what you see is that on a good year, maybe you have 100 fMRI papers in animals. And that's compared to the 1,000 MRI studies that come out in human neuroimaging every month. So I'm not claiming that we are anything as big as UK Biobank or anything as, you know, impactful at the moment, but we've been a growing community. And I think we have a potential. So here, I'm just showing you just to get you started. Two studies from our lab. So here we performed a literature meta-analysis for depression. It's just a quick way to see what is the current status of depression research. And we see that most studies represent hotspots associated with the disorder in this kind of singular area here, which is consistent with the findings that I've shown you from the original papers by Drevets. It's basically these kind of areas down there. And then if we apply a mouse model for depression using social stress, chronic social stress in mice, and we look at functional differences compared to control, we see it's the same single cortex that's affected as represented here. So basically, these mice spend more time in this network than control animals. And that's consistent with the concept of depression in humans. And basically that has the potential to try to understand the biological mechanisms behind this phenomenon. Similarly, for Alzheimer's disease, another example from a lab. Here we see, again, a literature meta-analysis for tauopathy, which is a pathological aspect that is found in Alzheimer's disease. And what we find is that there are literature hotspots always in the entorhinal cortex and associated ventral networks. And same concept, if we put animals that are developing tauopathy, the 3XTG mice at 3 and 6 months, and we look for functional differences within the brain, we see it's the same networks that are affected. And here on the side, you can see the development of the pathology where there's nothing in controls. And then in red, we're staining for tau, and this pathology develops over time. What does that tell us? It tells us, again, that this mouse model of tauopathy recapitulates functional deficits in the area where we'd expect them to be found in humans, and we can build on that to better understand the pathology and the functional consequences of that. So that would be good. And I'm really excited with these research directions, but I think we have to keep an eye open for the challenges that we are facing with functional MRI. In general, we're working at the detection thresholds. We are recording signal that is not directly linked to neuronal activity, so it's always complicated to interpret. Also hinted by the previous talk by Henning, for instance, where you could have all the confounding responses from the body and heart rate, for instance. Moreover, the signal that we acquire is confounded by multiple noise sources, so spiking from the scanner, drifts, also physiological noise sources like breathing and heart rate. The pre-processing is a nightmare at best and very complex in whenever, and that makes interpretation difficult. When we work with animals, it's made even worse, and that's why the effort by Henning are so important, because trying to get a synthetic understanding of all the variables that affect rodent imaging is a nightmare. We have to worry about maintaining the physiology. We always have the big debate versus awake animals versus anesthetized animals. If we go for the awake state, we are worried about stress. And the brains are much smaller, and we're also worried about signal to noise ratios. And, you know, it's just a complicated mess. So my approach has been somewhat different from Henning's, has been to call everyone in the community and ask them to give me their data, the way they acquire it. And so that led to a big study with 200 collaborators from 20 countries. We obtained, or I gathered, 82 data sets, each with n equals 10 rats that are representative of acquisitions around the world, for a total of 800 MRI scans. This is the map of the places where I have my collaborators, and I always use that as a way to invite new people. If you are people in the Midwest or on the West Coast in the US, if you are in Central Europe, or if you're in Asia in particular, I still have difficulties reaching out to these kind of labs. And if you are from these areas, and if you are working in this field, or if you know people who are, get them in touch with me. So the next studies, we can also include even more people from more places around the world. So first thing I like to do is give a brief descriptions of the data sets that we collected. So the first thing is the sex ratios, and it's always biased in science, unfortunately. And because we ask for data that comes from the labs, that same bias is also found in our study, in the sense that we have more males than we have females. People use a collection of different strains of rats, which is, again, consistent with the literature, with people liking the Wistar rats or the SD rats. People use also a lot of different anesthesia protocols, with isoflurane and medetomidine being the biggest contributors. And again, that links back to the previous discussion by Henning, how we use all different protocols and each of them having different impacts. And finally, we can also look at field strength, where a lot of people work at 7 Tesla or 9.4 Tesla, but we also have a wide range of other magnetic fields. And here, just to give you an idea of how difficult it was for me to run the study, is 3 different scans from 3 different rats, each having completely different contrast, especially here in the region near the amygdala. And you can extrapolate that to realize how difficult the pre-processing was, because the images were so different from one another. But we managed that. And what I want to discuss now is rather the big conceptual advance that we brought with our study. And the biggest advance, in my personal opinion, is getting to agree on what it is that we should be looking for when we're doing these kind of measurements. And the approach I've taken is to boil down the phenomena to its most simple form. So in task-free fMRI, we have a combination of temporal and spatial properties. First, with the temporal properties, here is a classical figure from Fox 2005 that everyone shows in the slides whenever they're talking about task-free imaging. Basically, what they're showing is, in hot colors, these are regions of the brain that are co-activated together, represented here by the orange and the yellow, which you can see if you look at the temporal signal, the signal goes up together and goes down together. And then they are also anti-correlated, and this can be symbolized by the blue here, where you see that the signal goes up in the blue region when it goes down in the orange and yellow, and vice versa. So basically, we can find brain regions that are co-activated together, and we expect that when some goes up, some regions are also deactivated. The other thing is that most of the pattern of coactivity happens with contralateral homotopic regions. So if the signal goes up in one region, it goes up in the contralateral region. And that's supported by the fact if you inject, as here, a viral tracer into the brain, it goes, usually the neurons project to the contralateral side. And this is also reflected in the neuronal network that we record with task-free fMRI. So using this 2 observations, we can sort of make a prediction. We look for strong correlations in the temporal signal between one brain region here and its contralateral part. And we also expect to have 0 or anti-correlations between that region of interest and a region of interest where we know that there's supposed to be anti-correlations as defined in all the other studies that have been done before. And here, this is the most important plot of our study. On the x-axis, we represent the correlation between 2 regions we expect to be correlated. And then on the y-axis, we represent the correlations between 2 brain regions where we don't expect to see correlations. And every dot represents a rat scan that was provided and has been color-coded by sequences. So when you do fMRI, you can either do gradient echo or spin echo EPIs, and here I've represented this way. And we created 4 categories for the scans. The first one being here, the scan at the center, which we call no functional connectivity scan. Let me represent them. So these are basically scans when we extract the time series from this one brain region, we see no apparent correlations either to the contralateral region or to that part in the center. And that's not something that you would want to have in your data, because functional connectivity is defined by these type of correlations. And about 30% of the scans that were provided to me fell into that category. So that's not great. The next category, we call it spurious correlations. We have correlations between our region of interest and that part where we said there shouldn't be correlations, but we don't have correlations with the contralateral sides, for instance. And collectively with this other category, which is called unspecific functional connectivity, where we have correlations between the 2 regions that we want, but we also have correlations with regions that we don't want to have correlated, which we call unspecific for that reason. So together, these two categories amounted to about 30% of the scans. And the point I'm making is that what we want to have is as much as possible scans in the last category, which we call specific, where we have strong correlations between the site of interest and the contralateral side, but actually no correlations or anti-correlations with this control region. And we had about 40% of the scan that fell into that category. And what that means is that on average, only about 40% of the scans that we collect contribute to the group signal that we represent in our maps, and 60% is either noise or random noise or just patterns that are not biologically possible. And that's a huge issue because it means there's a lot of data that does not contribute properly to your study. So with that information in hand, I called everyone from the study and I said, well, look, people, these are the kind of parameters that seems to have worked well on average in the best data sets. And one of them was the anesthesia protocol. So we took one anesthesia protocol from one of the beta study that we found. You can find all the references on GitHub and the paper, so it doesn't matter to look it up. And then we looked at sequence. And for sequence, people acquire things with a lot of different parameters. So here, my idea was to go for MRI sequence that works, as in a data set, that sequence had worked, but it also has to work for old system in a way that it can be inclusive for as many labs as possible. So this is a sequence that is used in a lab that operates a very old 4.7 Tesla scanner. So the idea is that if it ran on that old 4.7 Tesla scanner, it would run on every scanner. So it's not a fancy sequence. It doesn't do multiband. It doesn't do zero echo time, all of these fancy things that we see in the field. But the idea is this is a great place to start. So we discussed that protocol, and then I asked everyone to go back to the lab and acquire more data with it. And that's what they did. Oh, sorry. This one should not have been hidden. My bad. And this is actually the fantastic thing that happened is we got 21 labs that acquired data again, and then send me the data. And what we found was that there was a very large increase in scans in that specific connectivity category, 67% here. So basically what that means is if you were to do functional imaging task-free in rats, you could either take any random protocol that you find online, and then you would have about 40% chances of having good scans, or you can take our protocol and have 60 to 70% chances of having good scans. And that's a major difference because it will potentiate your data if you use our protocol, and will make sure that you have more robust results and you can have better inferences. But with every study that I do, there's also a lot of subtext, and the subtext here is that I want to promote open science. And the way I've done with it is first with pre-registrations. This is really, really hard to do, and not many of us do it yet, especially in the imaging community. I want to encourage it, and so that's why I've tried it out, and it worked for me so far. The second thing is the adoption of open source software that people can examine. And together with my friends at McGill University, we developed this fantastic set of tools called Rabies, which are fantastic for rodent fMRI pre-processing. The next thing is promoting open data and open code. So I'm terrible at coding Python, but you can actually go on our GitHub page and you can get all the code that we've made to make all of the figures in the paper. And if you think the code is good enough, feel free to use it. Same thing, all the data is available. And so if you download all the data and you run the code, you should be able to replicate the entire study. And finally, we've also came up with a vocabulary for quality control, quality assurance. And this is represented here by these plots. And so this actually is an output from the software Rabies that we have co-developed. And basically, we can agree what is the basic quality control, quality assurance that we should do to accept scan in our studies. And I'd like to finish with, again, this note about the current studies that I'm doing at the moment. A lot of people have been, we're trying to grow the kind of groundwork that we've done across multiple centers. So we're running a study at the moment with sensory evoked fMRI in rats. Henning is part of it. We almost closing it, but if you have data and you haven't contributed, it would be great to have you on board. We are doing a study dedicated on awake imaging in mice and rats. Again, for the reasons that Henning have pointed out, it's really hard to do. And if there are labs that are better at doing it than others, we would like to identify this and amplify this. We want to compare pre-processing pipeline. This is, again, something that everyone does separately. And we want to find what are the good pipelines that leads to better results. My friends in Toronto and EPFL have also been inspired by the study that we ran, and now they're running the same multi-center study for spectroscopy. And if you're interested, write to me, and then I would put you in contact with them. There's also a study on MRI quality control in Cologne. But the bigger question, and maybe something we need to discuss, and feel free to reach out to me if you want to have these kind of conversations, but it's really, what is the next big project that we need to run as a community? Something as important and, well, maybe not as big as UK Biobank, but something of that magnitude that needs to involve multiple labs, because it's such an important and big biological question that we cannot trust it to a single lab. So this is something we have to have discussions about, and I'm really excited to sort of start this thing and have discussions with people, so until we find like what are the cool projects to run. And with that, I have a huge number of people to thank, 208 to be precise. I'm not going to name them all, but I am in debt to all of them for the data, for the inputs, and the great ideas and the support. And with this, I'd like to thank you for your attention. And I'm very happy to discuss more about this project, either here via questions or by emails later on. Thank you.